Can you hear me?
Yeah, hi, it's Karis.
Hey, Karis, how are you?
Good, how are you, Antonio? How
Very
was
good.
Thanksgiving?
Very good, very good. Trying to recover.
Hello. Too much turkey.
Too much turkey.
Hi there, it's Kristen.
Hi, Kristen. Hi, Mustafa.
Yeah, we can see him. We can see you, Mustafa.
Yes, well, at least we can see his name.
Yeah.
Hi, can you hear me?
Yep.
Oh, good. Hi, everybody.
Hello.
Hello.
Hi, Antonio.
How are you?
I am okay.
Recovering from Thanksgiving.
Oh, yeah. Much recovery needed. So what's the order we are doing this in?
I think as usual.
Yeah, I'm just here to help with questions and anything like that. I'm certainly not presenting.
I know, Mustafa, I go after you. So I don't know if Kristen is first or last.
Okay. Hi, Miranda, are you on the call?
Yes, I am.
Will you be recording this for future use?
Yes, it's already started recording.
Okay. Well, let us know when we should start, I guess. It's 1 after right now. Kristen, when?
I'm ready whenever you are. It doesn't look like anyone else has joined.
Yeah,
I don't know
I see that.
Okay.
So wait a minute.
Just an FYI, I need to go to a meeting slightly before 4.
Okay.
So if I ring off, that's why.
going to post one more time. I posted again this morning about the call, but people may be coming off of the holiday busy.
Yeah, I suspect so.
I'm afraid.
We have 2 other people, I think.
Okay, good.
People are clear about the time, right? Eastern, Central, etc.
Yep, that's what it said on the post.
Okay.
Should we get started?
Sure.
Let's do it.
Okay.
Okay, thank you everyone for joining. First up, we have Dr. Mustafa Sahin from Boston Children's Hospital.
Thank you, Miranda. This is Mustafa Sahin. I'm a child neurologist at Boston Children's Hospital. I'm also the principal investigator of the Developmental Synaptopathies Consortium, which I'm going to tell you a little bit about with respect to the clinical trial on PTEN. Next slide, please. So we formed this Developmental Synaptopathies Consortium to study some genetic disorders that result in somewhat similar phenotypes in the patients. We call this consortium Developmental Synaptopathies to highlight the fact that we believe that many of these disorders are due to functional problems at a structure called the synapse, which is the structure which allows 2 nerve cells in the brain to connect to each other and to communicate. Synapses are specialized parts of the nerve cells, and a nerve cell connects and provides information to another nerve cell through these particular structures. And we believe that the dysfunction at the synaptic level is implicated in many neurological disorders that result in symptoms such as seizures, communication problems, social interaction problems, etc. Next slide, please. So the Developmental Synaptopathies Consortium was formed roughly 4 years ago, and it consists of 10 sites, and it focuses on 3 well-established genetic syndromes that are associated, each of them are associated with a high penetrant risk for autism spectrum disorder and intellectual disability. The 3 disorders are tuberous sclerosis complex, or TSC for short, PTEN hamartoma tumor syndrome, and Phelan-McDermott syndrome, or PMS for short. We believe that comparative investigation of these 3 disorders will allow us to identify common pathways that are disrupted in autism spectrum disorder and intellectual disability overall. Next slide, please. So if I may take a step back and look at the big picture in autism today, we believe that due to advances in genetics and genomics, there are approximately 500 to 1,000 different genes that give you susceptibility for developing autism. So this is a huge advance in terms of understanding the cause of autism, but this obviously causes a potential obstacle for developing treatments. One can think of several different scenarios to develop treatments. One might be a scenario where one broad spectrum intervention or drug may work for all genetic causes of autism. But due to our understanding of some of the genetic causes, such as Fragile X and tuberous sclerosis complex, we believe that these disorders may actually have very different pathologies. So what may work in one disorder may actually make the other disorder worse. So this idea of having a broad spectrum treatment for all forms of autism might not be so easy to achieve. On the other side of this spectrum, we can imagine a scenario where we have to develop a different intervention or a different drug for every genetic cause of autism. Again, this is a scenario that's extremely labor intensive and requires a huge amount of resources. So we hope we don't have to take that road to develop a different drug for each disorder. So many of us in the field are hoping that there might be subcategories of autism that might converge at the cellular level or circuit level, and developing interventions or drugs for these subcategories might have implications for a large portion of autism and intellectual disability. Next slide, please. So there are a number of goals of our consortium. Our long-term goal, as I mentioned, is to comprehensively characterize these 3 genetic disorders and to shed lights on the molecular pathways and circuits underlying autism spectrum intellectual disability. That's our long-term goal. But in the short term, what we hope to achieve is to characterize the developmental trajectory and phenotype of these 3 disorders by analyzing large subset of patients with each carrying mutations in one of these genes. And as a second short-term goal, we hope to identify biomarkers that predict risk for disease severity and disease progression. Next slide, please. So there are many partners in this effort. We are funded by 4 different institutes at the NIH. NINDS, NCAS, NIMH, and ICHG are the 4 institutes that provided funding for this study. We also have Novartis, a pharmaceutical company that's providing us the drug, and PTEN Research, a foundation in the United Kingdom that's providing partial funding for this clinical trial that we're doing. And we obviously work very closely with all the advocacy organizations, PTEN World, PTEN Life, PTEN Hamartoma Tumor Syndrome Foundation, and PTEN Research, as well as the other disease foundations for the other disorders. The project leaders are shown on this slide. For the overall study, I'm responsible as the principal investigator. On the PTEN project, Dr. Keris Eng, who's on the call, is the lead PI, and for the PTEN clinical trial, Dr. Antonio Harden, who's going to follow this presentation, is the lead PI. And we have a number of project and program scientists from NIH who are collaborating with us, as well as the Data Monitoring and Coordination Center at University of Florida. Next slide, please. So just a short review of the PTEN hamartoma syndrome and the PTEN mutations. PTEN stands for phosphatase and TENsin homolog gene. And as you probably all know, there are associated mutations in this gene are associated with a spectrum of clinical disorders that are characterized by both central nervous system and non-central nervous system manifestations. In the central nervous system, neurocognitive deficits, intellectual disability, autism symptoms are the main manifestations. Outside of the brain, this disease disorder represents with skin lesions, microcephaly, hamartomatous overgrowth of tissues, and increased risk of certain types of cancer. So PTEN turns out to play a crucial role in cells by inhibiting both the mTOR kinase pathway as well as the MAP kinase pathway. The reason we've studied PTEN hamartoma tumor syndrome with the other 2 disorders is that we believe mTOR pathway is afflicted in all of these 3 disorders, and the mTOR hyperactivity that we see in PTEN hamartoma tumor syndrome might be the reason for not only the cell growth and proliferation problems, but also for the central nervous system problems that we are studying in the Developmental Synaptopathies Consortium. Next slide, please. This is a cartoon manifestation of what I spoke about before, the mTOR pathway. So this is a cartoon of a synapse. On the top, the gray structure is the presynaptic terminal. So a nerve cell has sent out a connection to come to the synapse, and those blue vesicles, blue circles, are the synaptic vesicles by which neurotransmitter is released to the synapse. These bind to certain receptors on the postsynaptic side, this sort of light blue colored mushroom-shaped structure at the bottom. And the activation of those receptors leads to multiple signaling events in that postsynaptic cell, including activation of the kinase mTOR, mTOR can be inhibited by tuberous sclerosis genes, TSC1 and TSC2, or it can be inhibited by the PTEN protein. The SHANK3 mutation, which is the causative mutation in the Phelan-McDermid syndrome, seems to affect this pathway in the opposite way. So loss of SHANK3 actually results in loss of activity of mTOR. But we believe by studying these three disorders in comparison to each other will give us more information about how this pathway goes awry in the three disorders. Next slide, please. So the one advantage of studying mTOR in autism spectrum disorder and intellectual disability is that there are actually FDA-approved compounds that have been approved for other indications that can be repurposed to investigate this pathway either in the lab or in the clinic. One of those compounds is Everolimus, or known as Afinitor in the United States, and Votiba in the European Union. This is a drug that's made by Novartis Pharmaceuticals, and is the drug that we're using in this clinical trial, which you're going to hear about in more detail in a few minutes. It's basically an mTOR inhibitor, very similar to another compound that's been studied for decades in the lab and the clinic called rapamycin. And we believe, based on the animal studies and cell culture studies, that this group of compounds can improve the symptoms of physiological abnormalities in PTEN hamartoma syndrome associated with loss of the PTEN gene function. Next slide, please. This slide summarizes some of the studies my lab and others have done using mTOR inhibitors in the tuberous sclerosis disorder. We have shown that you can replicate some of the deficits and symptoms that we see in the patients with tuberous sclerosis in the mouse models. These include seizures, learning problems, and autism features. And each of these studies have shown that mTOR inhibitors such as rapamycin, or in some of them Everolimus, actually improve the symptoms. One of the first ones we did was back in 2008. We showed that we can improve seizures. Other groups showed that even if the seizures start, using rapamycin improves the frequency of the seizures. It reduces the frequency of the seizures. In a mild form of this disorder, you can improve the learning problems that these mice have. And more recently, in 2012, we showed that you can improve autistic-like features in the mouse model using rapamycin in the lab. So the basic question, obviously, is whether these effects that we're seeing can also occur in the clinical trial. Next slide, please. So looking at the history of this, rapamycin was isolated several decades ago. It works very specifically on a kinase called TOR or mTOR, which is actually named after rapamycin. It stands for target of rapamycin. And it was shown to shrink the tumors in the rat and mouse models of TSC. And then, as I showed you in the earlier slide, it was also found to reverse the neurocognitive effects in the mouse models. So what happened in tuberous sclerosis was that over the years, this led to clinical trials asking whether rapamycin or everolimus would improve the clinical symptoms in TSC patients. And it has now, everolimus itself is now approved for kidney tumors, brain tumors, and epilepsy for patients with tuberous sclerosis after all these clinical trials. The same question has not yet been asked in PTEN hamartoma syndrome. That's why we're doing the clinical trial for neurocognition in PTEN, and we started this in 2017. Next slide, please. So one may ask, why did we focus on neurocognition? One of the aspects is that individuals with PTEN have, in addition to the cancer manifestations, have several behavioral and cognitive features, and there have been no studies to date that we know of that have attempted to intervene for the neurobehavioral and neurocognitive aspects of this disorder. And we believe that mTOR inhibitors, in particular, in this case, everolimus, might actually improve these symptoms by regulating the mTOR pathway that is hyperactive in individuals carrying PTEN mutations. So we believe this is very reasonable hypothesis that just has to be tested in the best clinical trial design that we can come up with. So I'm going to turn the microphone over to Antonio Harden to give you more details of the research design for this particular clinical trial.
Okay. Thank you, Mustafa. Hi, my name is Antonio Harden. I'm a child psychiatrist here at Stanford University. It's been great being associated with the group and working with individuals with and families with PTEN mutation. I've been working in the field of autism and intellectual disability for 15 years, but I'm very excited about this trial because right now we are using a disease-specific agent that hopefully will help us target deficit that we see with individual with PTEN mutation, neurocognitive deficit that we see in individual with PTEN mutation. Previously, in the trial that we've conducted before, we were shooting a little bit in the dark. When we had an agent, we didn't have a disease-specific agent. Right now, it's a great opportunity to be able to use a compound that's been tested in animal models and showed that it can reverse some of the abnormalities observed in animal models and to be able to test it in individual with PTEN mutation. Next slide, please. Very good. So what are the aims of this trial? First thing first is the safety of everolimus. Even though everolimus has been approved in other conditions, we don't know if this medication is safe in patients with PTEN mutation. That's why when we start with a new compound, when we're studying in a specific population, we want to make sure that it is safe. And that's probably the main focus of the trial that we are conducting. So we want to look at the safety, how individuals with PTEN mutation are tolerating everolimus. However, in addition to the safety, we are looking at other objectives. We call them secondary objective. Whether this compound is effective in the treatment of neurocognitive deficits and some of the behavioral problems that we see in individuals with PTEN mutation. That's why we're casting a wide net with regard to the behaviors and the cognitive deficits that we are targeting. One, we are looking at working memory, we are working on processing speed and fine motor skills. And these domains have been found to be deficient in individuals with PTEN mutation. In addition to looking at these cognitive markers, we are looking at behaviors, we're looking at attention, concentration, social deficit, repetitive behaviors, and adaptive abilities. In addition to these cognitive and behavioral markers, we are looking at some biologic markers. We are looking at three different markers. One is eye tracking task. We want to see if everolimus will lead to changes in eye tracking of individuals with PTEN mutation. Also, we're looking at resting state, encephalograms, EEG, and auditory evoked potentials, because biomarkers might be a little bit more sensitive than behavioral and cognitive markers. In addition to the secondary objectives, we have an exploratory objective, which is basically trying to see whether molecules that have been linked to PTEN and that the PTEN associated pathway molecules will change with the treatment with everolimus. Also, we want to look at the gut microbiome and the microbiome to see if they will change with treatment. Next slide, please. So this is an overview of the trial. It's a phase 1, phase 2 trial, which is the first level of examination of a compound to see whether it is safe and effective in the treatment of a specific population. It's a double-blind randomized controlled trial. That's very important because open-label studies are not very informative. We chose to have 3 sites, and as I mentioned, we have a primary endpoint, which is the safety and the efficacy, and also we wanted to look at the effect on neurocognition after 6 months of treatment. Next slide, please. So we have 3 sites, and covering a little bit most of the US. We have on the East Coast, we have Boston Children. In the Midwest, it's Cleveland Clinic. On the West Coast, we have Stanford University. Next slide, please. So I will go briefly over the trial design with regard to patient flow, inclusion-exclusion criteria, the way how we designed the study, and the primary endpoint. We look at the efficacy composite index that we developed, and we'll cover a little bit some other related issues such as dosing and time of taking the medication during the day. Next slide, please. So what's the participant flow? The goal is to randomize about 40 individuals. 20 will go into the active arm, and it's, as I mentioned, a 6 months double-blind phase. And the 20 will be randomized to the Everolimus, and 20 will be randomized to the controlled arm, which is a placebo. At the end of the 6 months, individuals who were randomized to the active compound will exit the study. The individual who were randomized to the placebo arm will be offered and invited to be part of a 6 months open label phase, where we will use the same design that we did in the double-blind phase, but in an open label fashion. And as I mentioned, it's a 3-site trial. At Cleveland Clinic, we have Dr. Rabi Hanna, who is the PI, who is definitely the support of that, Dr. Karis Eng. And at Boston Children, we have Dr. Mustafa Sahin. And at Stanford University, we have a team that I am leading here. Next slide, please. So what are the inclusion criteria? The reason for having inclusion-exclusion criteria is to make sure that individuals who are participating in the study are eligible to participate, and more importantly, it is safe for them to participate in the study. So the age range is from 5 to 45 years of age. Definitely, we want to have a documentation of a PTEN mutation. The cognitive criteria is for individual to have a non-verbal IQ, of a verbal IQ, or even a full-scale IQ above 50 to be able to participate in the testing procedure. We want to make sure that participants who are eligible in the study, they are performing below the average on one of the 3 domains that we feel are the most impacted in individuals with PTEN mutation. The 3 domains are processing speed, working memory, and fine motor skills. And we have 3 different tests that we use to assess these domains. We use the continuous performance task to assess processing speed. We use the Stanford Binet to assess working memory, and we use the Purdue Pegboard to assess fine motor skills. Another inclusion criteria is making sure that individuals who are about to participate in the study, they have a stable medication for at least 2 months, because we don't want to make changes in the medications. And if there are any improvement in the trial, we want to make sure that the improvements are related to the compound that we're studying, and in this situation, Everolimus, and not related to other changes that are occurring. We want to make sure that the individual is healthy and have adequate bone marrow and liver function and kidney function. We want to make sure that individuals, especially females who are not pregnant or planning to become pregnant, and if someone has any medical problem, we want to make sure that the medical problem is stable and the problem is not active. For individuals who are anticipating any changes in their interventions, if they're planning a change in their seizure medications or in their psychotropic medications, we want to make sure that the individual don't participate before they change the medication. We want to make sure that they are stable with regard to these interventions before including them in the study. And because of the tests that we use, we want to the participant have to be fluent in English. Next slide, please. So what are the exclusion criteria? There is a long list of exclusionary criteria. Again, this is to make sure that individuals who are participating in the study can participate in the study, and it's safe for us to give them the compound. First is that we want to make sure that they are not sensitive to the drug. and they don't have gastrointestinal impairment that will affect the absorption of the drug. We want to make sure that patients who are not receiving anti-cancer therapies, or if they received anti-cancer therapies, it's not within the last 4 weeks before starting everolimus. Because of the potential medication interactions, there are some medications that cannot be used at the same time as everolimus, and one of these medications, medication that affect the cytochrome P450, or they are strong inhibitor or inducers of this pathway in the liver. As I mentioned in the inclusion criteria, women of childbearing potential must agree to use highly effective birth control methods, and male participants that are of childbearing potential must agree to use adequate contraception as well. Individuals who have active infection at the time of enrollment will not be included as well. Next slide, please. So, again, individuals with significant medical illnesses such as diabetes mellitus, uncontrolled diabetes, uncontrolled hyperlipidemias, individuals who are receiving corticosteroids or immunosuppressive agents also will not be included in the study. Individuals who have history of HIV, individuals who were recently participating in a clinical trial also will not be included. The individuals who were recently in a clinical trial, they have to wait 30 days before entering in this study. Patients who have received live attenuated vaccine within 1 week of start of everolimus and during the study will be excluded. Most IM injection vaccines are not live attenuated vaccines, so most people will be okay. Patients who have history of another primary cancer will be excluded from the study and cannot be eligible for the study. Individuals who have had major surgeries or neurosurgery within prior 6 months of enrollment cannot be eligible for the study. Next slide, please. Okay. Everolimus administration. So when an individual is eligible to be part of the study and we did the screening phase and they are ready to go, once we randomize, we give them the medication, and these are the recommendations. We suggest to take the medication at the same time every morning after a low-fat meal with a glass of water, and pills should not be crushed or dissolved. If an individual misses a dose, that should take the dose as soon as possible within 2 hours of the typical dosing time. And if that's not possible, then they will have, fortunately, will have to skip the dose for that day. And usually we don't recommend taking 2 doses on the same day. Next slide, please. What are the potential side effects of everolimus? You know, everolimus has been used quite a bit, and we have quite a lot of experience using it. But as I mentioned, we don't know how individuals with PTEN mutation will respond to this medication. What we know about the side effects of everolimus is that the majority of these adverse events are mild to moderate. I'm going to list some of the side effects that we encounter. It does not mean that individual who will be taking everolimus will experience these side effects, but it is possible that they might experience these side effects. So the side effects that we see are mild and transient, such as fatigue, headache, gastrointestinal events, peripheral edema, headaches, nosebleed, mucosal inflammation, weight loss, respiratory symptoms, sometimes cough and dyspnea, shortness of breath, infection, sexual dysfunction, and skin conditions. Sometimes we see allergic interstitial pneumonitis, and at times we see psychiatric disorders or psychiatric symptoms such as insomnia and very, very, very rarely aggression. Next slide, please. In addition to the physical symptoms, we see abnormalities in the laboratory test. We see a decrease in hematology parameters such as hemoglobin, lymphocytes, platelets, and neutrophil, and that's why in the study we monitor blood work on a regular basis. We also see increased level of cholesterol, triglyceride, glucose, liver function tests, creatinine, and bilirubin. We saw also decreased level of phosphate and potassium, and we also see some level of renal dysfunction and proteinuria. And that's why we have very rigid schedule with regard to monitoring any of these side effects. Next slide, please. Additional study information, additional things we do at the screening. We want to make sure that individuals don't have HIV and they are hepatitis B and hepatitis C negative. The visit, usually we have a window which is plus minus 14 days. A lot of things can happen. Sometimes individual, there is Thanksgiving or there is the holiday break. So we want to maybe flexible to families who are participating in the study. That's why there is a window, but the window cannot be too long. That's why we like the visit windows to be within 14 days. Randomization is 1-to-1. What it means is that 50% of the participants will be randomized to the active group and 50% will be randomized to the control group. The dosing is usually 5 milligrams or 2.5 milligrams, and we can at times give it every other days depending on the weight of the individual and the height of the individual. We are targeting a blood level of 5 to 15 nanogram, and that's why for individuals who are in the study, we do what we call a PK levels, which means trying to measure the blood level of everolimus, and this helps guide us with regard to the exact dose and the most safe dose for these specific individuals. So those adjustments may also be made based on side effects, not only based on the blood levels. And as I mentioned earlier, the main goal of the study is to make sure that this compound is safe in the individual with PTEN mutation. That's why our threshold is very low with regard to side effects. Also, since it's a multi-site trial and we are using scales that are being administered in 3 different sites, we develop a training process, and we make sure that the raters are reliable between the 3 sites. Next slide, please. Okay, where are we today? So as Dr. Sahin mentioned, we started recruitment at the end of 2017. So it's been a very active 11 months. And today we have 24 participants who were screened. 7 individuals were screen failure, which means they did not meet inclusion and exclusion criteria. We have 2 eligible individuals who we felt that they will be able to participate in the study and decided not to. One of them is because the child was experiencing significant medical problems and the parents preferred to not participate in the study because too much was going on. We have 15 eligible who were randomized. 2 completed the trial, the double-blind and the open-label phase. We have 9 individuals in the double-blind phase at this time. We have 3 individuals in the open-label phase, and 1 individual who was randomized but decided to withdraw after randomization. So as you see, we've been very active, and the good thing is that we are seeing mild side effects. We are not seeing any unusual side effects, which is somewhat similar to what's been reported before. But final conclusion about side effects cannot be made before the study is complete. Next slide, please. Okay. Now, who are the people who are making this possible? At Boston Children, we have Dr. Srivastava and Dr. Sahin, as well as Raina and Mia. I see Mia is on the phone and Dr. Sahin is on the phone. Next slide, please. At Stanford, there is Dr. Phillips and Dr. Fung, as well as 3 coordinator, Robin Libov, Stephanie Gassan, and Jaylene Edwards. Next slide, please. And at the Cleveland Clinic, you have the largest investigators, we have Dr. Ang and Dr. Hanna. We have Dr. Tamburo, Dr. Sullivan, and Dr. Ashelman, and Dr. Flagg, and Dr. King. And we have Kamisha also, who's the main coordinator. And we have Kathy Balak, who's the pediatric nurse, and Dr. Rotz and Robert Hartley, the study coordinator. So as you know, it takes a village literally to do this trial, and this is definitely one of the most complex trial that I've been part of. Very good. Next slide. Very good. Thank you, and I will transition to Kristen, who will be taking over and finishing this webinar.
You can just say Hey, this is Kristen. I think this is not my slide.
I'm happy to give you slides if you want, Kristen. I think there is a couple of slides on PTEN. Can we have the next slide, please?
Yeah.
Oh, this is for the PTEN Research Foundation, and this is about their mission. As you can see, they were established a year ago, and they're supporting some of the research ongoing in this consortium. Next slide, please. And these are the people involved in that consortium, in that foundation. Riccardo Zacconi is the founder, and there are a number of scientific advisory board members, some from US, some from European Union and other places. Next slide, please. And this is, slide didn't come out well for some reason, but basically it shows that they're interested in both clinical development as well as preclinical evidence and targets to take on the various manifestations of the disorder. Next slide, please. So they have been supporting research by awarding grant support in basic translational and clinical research, supporting this particular trial, which we just talked about, engaging in pharma and regulators, and supporting the PTEN hamartoma syndrome community as well. I think that's the last slide. I'll turn it over to Kristen now.
All right. Hi, I'm Kristen, president of the PTEN Foundation in the United States. And next slide, please. I'm just going to give you a highlight of some of our projects that we've done over the past months. We successfully launched our first International Family Council, and the council worked together very successfully to host our first International Online Awareness Day, which was on October the 23rd, 2018. It was a huge success. I know just from our Facebook statistics, we had over 25,000 people that were reached, which is pretty incredible. And in large part, it's because of our awesome family council. And I had to give a shout out to the Cleveland Clinic team. They were phenomenal in joining us in this effort. You can see just one of the pictures that they put together in the community was just really ecstatic about this participation. So thank you. Next slide, please. Okay, we recently launched our Centers of Excellence application, which can be found on our website. We're really excited about that, and it was a collaboration with our Scientific Advisory Board. Both Dr. Sahin and Dr. Ng have participated in that effort. We thank them very much for that help. We also launched our first PTEN Awareness Public Service Announcement commercial teaser. We did this on our Awareness Day, and the full commercial, which is really incredible, should be out this week, so look for that. Please save the date and mark your calendars for our second annual Patient and Scientific Symposium, which will be held at the Cleveland Clinic this year on April 7th and 8th. I believe I have my dates right there. 2018, or 19 actually, sorry. And then we will be launching before December the 31st our first effort at a patient-powered registry, and we are very excited about that as well. Next slide, please. Okay, do you want me, some of these questions were addressed very well, so thank you, Dr. Harden and Dr. Sahin for that. But we'll go through just for those who took time to submit these and maybe touch on them just briefly. First question, as you can see, is the trial more about safety of the drug in humans? And would you actually be able to see cognitive improvement and in fine gross motor skills in 6 short months? Do you want to just do one by one? Is that okay?
Sure.
Okay.
I can take.
Okay, go, Mustafa.
Okay. So obviously, at this stage of drug testing, our highest priority is always in the safety. We have 2 primary goals. One is the safety, and that is utmost for us. We want to make sure there are no significant major side effects. All drugs, as you know, have some side effects, but we want to make sure that if there are any side effects, they are reversible and tolerable. But we are also looking for the efficacy signal. You know, to be able to move to a later stage trial, we want to see at least that some of the patients are benefiting, and we do hope 6 months is sufficient enough time to see some effects. This is partially based on the pace in which we can see improvements in the preclinical studies. That is, studies we've done with animal models. We've been able to see effects within a few weeks of drug administration. Now, you know, obviously we realize that mouse and human experiments are not the same, but we thought that 6 months was a reasonable amount of time.
Yeah, I can add that I think it's a good question whether 6 months will be enough or not. But the something that we have to keep in mind is a practical trial. Usually when trials are longer than 6 months, even longer than 4 months, the attrition rate is very high. That's why 6 months we felt it's a good, it's a reasonable number of duration that will allow us to see some benefits. The other thing with regard to the duration, it might vary depending on the age of the individual. So it's possible that young individual might benefit a little bit in a shorter duration compared to older individuals. These things we'll definitely be looking at once we have the results of the trial.
And if I may jump in, if the underlying abnormality has some metabolic component versus a physical anatomic component, then we will see the reversal very quickly, even within a couple of weeks.
Mm-hmm.
Fantastic. Anybody else on that one? No? Okay, second question. If the trial goes well, how quickly would it be available, I'm assuming the drug, and how long would it take to be available in Canada? And if the drug, I'm assuming if it comes on label, what would be the next steps to getting this drug or to help bring it on label?
I think I can take, I think it can become available as soon as the study is the finding of the study are the finding of the study are published, because then everyone will become aware of it. As we all know, this study is the compound is already available in the US, and if there is enough scientific data to support its use, then practitioners might be comfortable prescribing it. Now, the study that we are doing is a pilot study. It's a small study. It will provide a signal. Ideally, we want additional studies to confirm our findings if our findings are positive. So that's with regard to the first question. Now, I'm not sure if it's available in Canada. I don't know if Mustafa, if if everolimus is available in Canada?
I believe so, but I'm not certain. I don't prescribe there, so I'll have to check on that.
Yeah. I'll be surprised if it's not, but that's the. Usually they go hand in hand in terms of the U.S. and Canada.
And just to add on to what Antonio was saying, for drugs to be covered by insurance, a lot of times FDA approval as an indication is required. So that's why we're going through these phases of trying to prove that it is efficacious and safe for this indication, and that's the level of evidence we need for the FDA to approve this drug for this indication, and then most of the time, once the FDA approves it, it gets covered by insurance.
And that's
this is Rob. I just want to confirm that it is actually approved in Canada. It is the same product that it is in the US under the name of Afinitor.
Great, thanks, Rob.
Thank you.
Okay, next question. How does everolimus affect fertility? And is it a long-term effect?
So I can sort of take on that. There was a recent study done by the tuberous sclerosis researchers and was published about a year ago, looking at infertility problems with everolimus, and they basically show that there might be some endocrine dysfunction and menstruation issues raised in tuberous sclerosis patients taking everolimus, but there were no major safety concerns, and they all seem to resolve. So it's a relatively large study, about over 100 patients. So I think it's overall well tolerated, even though there might be some menstrual cycle problems, amenorrhea being the more common one. They seem to resolve and they are not long-term.
Okay, fantastic. You guys touched on this earlier, but maybe you just want to highlight one or two negative side effects and perhaps a positive side effect or a positive effect that you've seen. You don't really have to go into a deep dive because you did that earlier.
I think in the current trial, and as I mentioned, we're not seeing anything unusual that has not been reported. And now, one of the challenge is we are in the double-blind phase, and most participants are in double-blind phase, and it's very hard to tease out whether the side effects that are being observed are related to the drug or coincidental related to the placebo. But my impression, and Dr. Hanna and Sahin can weigh in, is that we're seeing very similar to what we observed in the other conditions, and we're seeing mouth sores and stomatitis and fatigue, and sometimes sleep disturbances, and sometimes some individual, their blood, their hematology is being affected. With regard to the positive effects, I don't know if Mustafa, you want to report on the case that you guys saw at Boston?
Sure. This is just an anecdotal case, obviously, but one of the individuals that took part in the trial seemed to have significant improvements in social abilities as well as language-wise. So this was reported by his parents. So the trial is ongoing. Obviously, we need many more subjects to finish the trial to be able to draw any conclusions. But it seems to be quite well tolerated. That's the main thing we can say. As I mentioned earlier, the tolerability and safety is our major concern. And from that perspective, this trial is going very well, with a few patients, with a few individuals reporting positive effects so far.
Okay. On to question 6, as parents considering this trial for our child, what would the top considerations be? Maybe just the top 2 as they're trying to make a decision.
I can say, and I think the main thing that parents will have to think is regarding if their child is medically stable to be able to be part of that the study or not. And the secondary one is ability to time-wise to be able to come to one of the sites to be able to participate. So the first thing is basically the safety, the medical stability of the child.
Okay, fantastic. And the last question, which you guys also touched on very well, excluding factors for this trial and why. And I'll give you one specific that a parent brought up to me. The child was non-verbal and thus excluded them from the trial. The parent asked if I can translate or or work through this with my child, would that make him or her eligible?
Yeah, as I mentioned, the primary outcome measure is tests related to neurocognitive functioning. And the best way to assess neurocognitive functioning is direct assessment with the child. So that's why, unfortunately, for some individuals who are minimally verbal, they might not be able to do these tests.
Okay. Answers? I think that's all the questions that were submitted to us. And thank you all for providing this for our community and giving us so much detail and for everything that you guys do on a daily basis. I know I speak for the community in saying that it's much appreciated.
There's one question on the chat. I think maybe we can address it before closing up. The question from ZL. Metabolic differences may be affected more quickly than structural. Karis, are you still on the call?
Karis had to drop off.
Oh, okay.
It looks like they were discussing metabolic chemical imbalance versus structural changes to the brain. And ZL asked a follow-up question about whether metabolic differences may be affected more quickly than structural differences.
Yeah, I will sort of, I guess, speak for Karis. I hope I'm not stating her what she was saying. But basically, what we think of is that if there are structural lesions, like we see in tuberous sclerosis, or sometimes, some MRI abnormalities that we see in individuals with PTEN, they may not go away within a short period of time. But we think that the synaptic communication might be easier to normalize, and that's, I think, one of the things that she meant by metabolic differences, that neurotransmitter, for instance, responses or secretion might be recovered quicker than the structural abnormalities that we may see on the MRI or at the microscopic level even. I don't know if that answers your question. Okay, thank you. Any other questions? If not, we want to thank you again for taking the time to take part in the webinar. I hope we've answered your questions. And thanks for being interested in this really important clinical trial.
Thank you. Thank you, guys.
Thank you.
Bye-bye.